colony can appear entirely healthy on the day it begins to fail. The bees are flying, the hive is buzzing, and the frames look full, and yet inside, something has already shifted. The colony has already begun responding to a threat that the beekeeper cannot yet see. This is not a failure of observation. This is a fundamental feature of how colonies process and communicate disease, and it operates weeks before any visible symptom appears. Have you experienced this in your own hives? A colony that seemed strong right up until it didn't. The biology of pre-symptomatic colony behavior is one of the most consequential and most overlooked areas in practical beekeeping. If this kind of evidence-based breakdown is useful to you, a like helps this content reach more beekeepers who are asking the same questions. For much of beekeeping history, disease management was reactive. A beekeeper would open a hive, identify visual symptoms, sunken cappings, discolored larvae, abnormal brood patterns, and then make a decision. This model of inspection has dominated practical beekeeping for over a century, but it rests on an assumption that has never been fully examined, that the first visible symptom is the beginning of the problem. It is not. What early observers understood but rarely articulated was that behavioral changes preceded physical ones. Experienced beekeepers in the late 19th and early 20th centuries noted that colonies about to show disease often changed the way they moved, the way they guarded, the way they responded to disturbance. These observations were recorded in apicultural journals and inspection notes, but they were rarely formalized as a diagnostic framework. They remained in the category of beekeeper intuition, the kind of knowledge passed through apprenticeship rather than textbooks. What those early observers were witnessing was the colony's social immune system in its early activation phase, and understanding that phase changes everything about how and when a beekeeper should intervene. To understand what a healthy colony does before it shows disease symptoms, it is necessary to understand that a honeybee colony does not have immunity the way a single organism does. There is no central immune organ, no unified biological commander deciding how to respond. Instead, the colony operates through distributed social immunity, a network of overlapping behaviors performed by individual bees that together constitute a colony-level defense. This mechanism has several components. Hygienic behavior, the detection, uncapping, and removal of diseased or dead brood is the most studied. Grooming behavior targets external parasites. Propolis deployment modifies the microbial environment inside the nest. Guard bee activity regulates what enters the hive, and foraging pattern itself reflects energetic allocation, the colony's biological assessment of whether it has the surplus capacity to maintain both production and defense simultaneously. These behaviors do not operate in isolation. They shift together, in patterns, in response to pathogen pressure, and they begin shifting before the pathogen has caused enough damage to produce a visible symptom. Full frames, empty prognosis. This is the core tension of pre-symptomatic colony assessment. A frame that looks healthy to the eye may already be showing, to the colony, a signal that has triggered a cascade of behavioral adjustment. The beekeeper sees full capped brood. The colony has already begun the process of responding to what lies underneath. Here is where timing becomes critical. The window between first social immune activation and first visible symptom is not a long one. Depending on the pathogen, whether bacterial, fungal, or parasitic, this window may be as short as 10 to 14 days or as long as 3 to 4 weeks. Within that window, the colony is doing work. It is investing energy in defense. It is allocating bees to hygienic tasks rather than foraging. It is changing its own internal economy in ways the beekeeper does not yet see. If the beekeeper misses this window, the intervention that was manageable at day 7 becomes difficult by day 21 and may be insufficient by day 35. This is not a problem of products or treatments. It is a problem of observation timing. The hygienic behavior mechanism is worth examining carefully because it is the most measurable of the pre-symptomatic indicators. Worker bees capable of hygienic response possess the ability to detect chemical signals from diseased or dead brood through sealed cappings. Research into this behavior, much of which builds on work conducted through the mid-20th century, has established that this detection is olfactory, mediated by volatile compounds released by compromised larvae. What this means practically is that the colony begins uncapping and recapping brood before the beekeeper can identify any visual brood abnormality. A beekeeper running a standard inspection might see a spotty brood pattern and classify it as a laying issue, a queen problem, or simple variation. But a spotty brood pattern in a colony with otherwise strong population metrics is frequently a sign of active hygienic behavior, the colony removing compromised brood and the queen attempting to refill those cells. Active bees. Compromised brood. The misidentification of hygienic activity as a queen problem is one of the most common and consequential errors in practical hive assessment. It leads to queen replacement in colonies that are actually functioning at a high immunological level. The replacement disrupts the brood cycle, removes the existing queen's established egg-laying rhythm, and often reduces the colony's capacity to manage the underlying pressure that triggered hygienic behavior in the first place. What most beekeepers misunderstand is this. A spotty brood frame in an otherwise strong colony is more likely evidence of immune function than evidence of laying failure. The distinction requires context, population size, forager return rate, propolis distribution, and guard behavior at the entrance, but it is a distinction that changes the beekeeper's entire response. If you value biology-based beekeeping, consider subscribing. Beyond brood pattern, the colony communicates pre-symptomatic disease pressure through foraging behavior. This is less frequently discussed, but it is well supported by observational research and consistent with historical beekeeper records. Colonies under pathogen pressure shift their foraging allocation. Specifically, they increase collection of antimicrobial plant resins, the raw material for propolis relative to nectar and pollen. This shift is subtle. It does not produce an immediate measurable drop in honey production, but it represents a reallocation of foraging energy from surplus production toward nest defense. Early 20th century beekeepers who kept detailed hive weight logs sometimes noted this pattern without fully understanding its mechanism. A slight plateauing of hive weight during what should have been a strong flow period, followed weeks later by the appearance of disease symptoms. They often attributed the weight plateau to weather or forage variability. In some cases it was, but in others, the colony had already shifted its economy inward before the beekeeper had any clinical evidence to act on. The practical application of this biological understanding begins with redefining what a standard hive inspection is designed to detect. A conventional inspection focused on visible disease symptoms, clinical foul brood signs, visible mite loads, discolored larvae, is a late-stage assessment tool. It is valuable. It should not be abandoned, but it should be preceded by a behavioral inspection layer that occurs before the frames are ever pulled. Before opening a hive, a beekeeper working within this framework examines 4 observable pre-symptomatic indicators. First, entrance activity. Not simply the volume of bees, but the quality of returning forager behavior. Foragers returning with large resin loads on their corbiculae, disproportionate to the season and available forage, signal increased propolis collection. Second, guard behavior, the response of guard bees to approach and low-level vibration. A colony under immune stress often shows guard bees that are either hyper-responsive, agitated at thresholds that would not normally trigger alarm, or in more advanced cases under-responsive, suggesting a compromised population structure. Third, flight pattern, whether foragers are returning on direct paths or showing erratic low-altitude circling, which can suggest navigational disruption associated with specific pathogen loads. Fourth, hive sound. The acoustic signature of a colony changes measurably under stress, though this requires a trained ear and is the most subjective of the 4 indicators. These 4 observations take fewer than 3 minutes and require no tools. They precede the visual frame inspection. They do not replace it. Once the hive is opened, the pre-symptomatic assessment continues with what might be called a frame economy audit. This is not a search for clinical symptoms. It is a population and allocation assessment. The beekeeper notes the ratio of bees on brood frames to bees on honey frames, the density of bees per frame, rather than simply the count of covered frames, the continuity and geometry of the brood nest, and the distribution and freshness of propolis deposits. Propolis deposits, particularly in the corners of frames and around the inner cover, serve as a passive record of the colony's recent defensive activity. Fresh, soft propolis in areas that previously had little indicates recent deployment. This is a behavioral artifact the colony leaves behind that the beekeeper can read across successive inspections. Temperature inside the brood nest should be maintained between 93 and 95 degrees Fahrenheit under normal conditions. Colonies under significant pathogen pressure sometimes show slightly irregular heat distribution across the brood nest, warmer at the margins, variable at the center, reflecting uneven nurse bee coverage. This requires a calibrated thermometer and multiple readings across the brood frame surface, but it provides an early physiological indicator that precedes visible brood abnormality. It is the kind of measurement that takes practice to interpret correctly, but once understood, it narrows the beekeeper's assessment window considerably. The decision window framing matters here. A colony assessed at day 7 of immune activation. When propolis collection has increased, guard response is slightly elevated, and a small number of cells show hygienic uncapping, presents a manageable situation. The colony's own immune architecture is functional. The beekeeper's role at this stage is primarily to reduce additional stresses, avoid unnecessary inspections that chill the brood, ensure adequate protein stores to sustain nurse bee populations, and assess whether the colony has the population density to maintain brood temperature during cool nights. At day 21, the picture has changed. Hygienic behavior has been sustained at an energetic cost. If the pathogen pressure has not been reduced by the colony's own mechanisms, the colony has been drawing on reserves to sustain immune function. Nurse bee populations may have fallen below the threshold needed to adequately cover the brood nest, which means brood temperature regulation becomes inconsistent, which means brood development becomes irregular, which introduces additional stress. The cascade becomes self-reinforcing. At day 35, the clinical symptoms the beekeeper feared have arrived. The intervention window has narrowed from wide to narrow to difficult. Seasonal timing intersects with this framework at every point. The pre-symptomatic behavioral signals are hardest to interpret during early spring, when colony population is rebuilding and brood nest geometry is naturally variable, and during late summer, when colony population is contracting and forager ratios shift as winter preparation begins. Both of these transitional periods can produce behavioral patterns that superficially resemble pre-symptomatic immune activation. The distinguishing factor is trajectory. A colony in normal spring buildup shows increasing forager return rates, an expanding brood nest area, and decreasing propolis activity as the season warms. A colony in early spring that shows stable or declining forager return, contracted brood geometry, and increasing propolis activity is not showing spring transition behavior. It is showing immune activation during a period when its population is at its lowest annual point, which makes the intervention window both more critical and more fragile. Late summer contraction produces the mirror problem. A colony reducing its forager population in preparation for winter may show reduced forager return rates and a contracting brood nest that mimics immune suppression. The distinguishing indicator here is the presence or absence of increased resin collection. Winter preparation does not produce a resin collection spike. Immune activation does. Colonies that move through an undetected pre-symptomatic phase into clinical disease during late summer or early fall face a compounding problem. The bees that survive a disease event in August and September are the bees that will become the winter cluster. If those bees have experienced extended immune system activation, sustaining hygienic behavior, grooming, and propolis deployment at elevated levels for weeks, they carry shorter functional lifespans than bees that have not been under sustained pathogen pressure. Their hypopharyngeal glands, which produce the brood food necessary to raise healthy winter bees, may already be partially depleted. The winter cluster they form will be smaller, less vigorous, and beginning the spring buildup from a lower biological baseline. This is how undetected pre-symptomatic disease becomes a spring loss. The colony does not die in September. It dies in February or March. And the connection between the late summer immune event and the late winter failure is not always made. A beekeeper who learns to read pre-symptomatic behavioral indicators does not achieve disease prevention in an absolute sense. Pathogens are part of the biological environment. Honeybee colonies have coexisted with for millions of years. What changes is the timing of awareness. Awareness at day 7 rather than day 21 does not guarantee a different outcome in every case, but it consistently shifts the probability toward outcomes where the colony's own immune mechanisms remain sufficient, where the beekeeper's supportive interventions are still possible, and where clinical disease is either avoided or managed before it becomes catastrophic. This is the realistic outcome of biology-based early assessment. Not a cure, not a guarantee, a stabilization of surplus, prevention of avoidable loss, and restoration of the colony to functional state before function has been severely compromised. A narrowing of the uncertainty window that the beekeeper operates within, which is, practically speaking, the most a careful observer can ask for. The bees have been doing this work long before beekeepers learned to read it. The hygienic response, the propolis deployment, the foraging reallocation, these are ancient mechanisms. They predate modern apiculture by millions of years. What is relatively new is the beekeeper's ability to observe them with enough precision to act within the window they create. A colony showing no visible disease symptoms is not necessarily a healthy colony. It may be a colony actively fighting for its health. The difference between those two states is measurable. It requires patience, a trained eye, and a willingness to look at behavior rather than only at clinical signs. The frame that looks fine may already be speaking. If this breakdown was useful, leave a comment with your experience. Subscribe for more historically grounded beekeeping insights.